Well, today we're going to be doing a kind of a little workshop for reading papers for like the very beginner level. We're going to take it real slow and walk through like everything that they did. This is it's a fairly, I'm not going to say it's easy, but it's definitely beginner friendly, this paper. Um, so we're going to cover a little bit about what makes an academic paper first and how kind of you read them, and then we're going to start reading this paper together and discussing it as we go. Okay. So the first thing I always like to start with whenever when I was an undergrad and I led every single presentation and meeting with this slide. These are the tools that I use for reading papers. Zotero is just an amazing kind of indexing source, and it's a PDF reader, so it lets you highlight things. It's honestly an amazing resource. There's a plugin so that you can just click a single button and it'll import papers from the internet. That's great. Research Rabbit is something I found really recently. You can sync it with Zotero, and then it will use AI to find relevant papers that like were cited or cited the paper that you already have in your library. So I use that to find new papers all the time. PubMed is great because it just has probably the most free papers besides for bioRxiv anywhere. So those are really the things I recommend if you're just wanting to get practice or if you're specifically looking at a topic and just want to kind of learn more about it. Okay, so the first thing we have to talk about for people that are new to reading academic papers is that scientific papers are really stories. The authors are trying to tell you something. They always have this three act arc that normal like dramas or any other kinds of books will have, where you start with the problem and then you're going to get from the problem to the main theme. That's going to be like the hypothesis, and you get there with all the background. That's what you're going to find in the introduction. And then you're going to have the results that are going to get you to the discussion. And the discussion is going to get you back to a new problem. That's usually where we find like the future directions or that kind of thing. So you really have this cyclical kind of nature where you have the problem, you find things about it, and then those things are going to inform future questions where we can find like new gaps in our knowledge. Okay, there are five main parts of an academic paper. Those are going to be the abstract, the introduction, the methods, results, and discussion. We're going to get started with the abstract first. So the abstract is really going to be a short summary of the paper, and it's important to get a roadmap and like hints about what's going to be important. Usually that's where you'll find all the buzzwords. It will tell you just in general what their methods are, what their results are, and it usually has some kind of like discussion or author's interpretation, just a little blurb at the end. So if you guys want to go ahead and pull up the paper that we're talking about today, I want you to read the abstract and highlight all of the following, or you can just make a note of them. I know, right? I said it's the galaxy cat for the abstract is I picked that one purposely because the abstract is kind of like the entire paper, like it's the whole universe. Too much effort. There's no such thing as too much effort. Yeah, I can send you the presentation. Okay, I'm gonna give you guys like 5 minutes to read the abstract and kind of make note of those things, and then we are going to kind of talk about it a little bit. Um, Participation is really important today. It is a workshop. I'm not just giving a presentation. Sona, it is right here. One thing I do want to touch on I picked this paper because it has a lot of jargon in it, but that jargon is ultimately kind of unimportant to understanding the paper. So if you come across a word that you don't understand, I want you to make note of that word, and then we're going to cover all of the words that people don't understand and kind of craft a better understanding of what's going on in this paper before we move on. Send me a friend request. Okay. I use Notion most of the time, pros. I Hi, Wheeze. I like the organization of Notion better. Like I have tables that'll like I can sort to find specific papers. Like I have papers that I've just started or that I've done like my first pass, second pass, third pass. And it lets me sort all of those. And it also lets me sort it by like topic. And I haven't really found Notion like has a good way to do that. But like I do keep notes while I'm reading the paper in Notion and then just transfer, or while I'm reading the paper, I keep notes in Zotero and then just transfer them over to Notion. If you're associated with, oh, the abstract. Okay, so hang on, what PDF did I send? There's a couple of versions that float around. Oh, okay. Um, so the abstract is, oh, you can't see that. Stop. So the abstract is going to be this section right here, that section. Bup? What is bup? Oh, okay, let me actually read the abstract. Okay, let's go ahead and get started talking about the abstract. So in other sections, we're kind of going to discuss, I'm going to ask for input, but for this section, I'm just going to show you what I have, and then you can kind of compare that to what you had. So this is what I really thought was important when I was reading this paper. First of all is the results. I'm always looking for the results first. That's going to be, I usually highlight results in purple. So creating a false memory is important. That's like the entire focus of their paper. That's why they did this. They were trying to create a false memory. And then there's increased freezing in the original context in which a foot shock was never delivered. And that this recall was context specific and activated the same regions that would have happened if it was a regular fear memory. This really is going to be the author's interpretation. It's it's basically something that they're extrapolating on. They're just like kind of hand waving a little bit. It's an educated hand wave, but it's not like data, right? And then this is going to be all of the kind of methods that they use is right here. Where's my pointer? There's my laser pointer. Is right here in yellow. So that's kind of how I went through this. This is what I thought was important, and these are really things that we're going to keep in mind as we kind of move on through the paper, especially when we talk about the results. So, does anybody have any words specifically in red, any jargon that they didn't know that they want to define before we move on? Optogenetics, yeah. Ingram, okay. So optogenetics and Ingram, those are the ones that I really expected. And then also dentate gyrus and CA1, maybe channel rhodopsin 2. You're in luck because those are all things that we're going to talk about. If the gene is expressed, we will also talk about that. Okay, let's move on a little bit. So the introduction is going to primarily be important prior research, and it's also going to include the central question or hypothesis, basically the entire reason why they're doing this paper in the first place. Um, this paper is a little weird because it's kind of a like an op-ed, I'd say. It's not formatted the way you'd normally see an academic paper. So for this paper, the first two paragraphs are going to be the important prior research. But before we go through that, we're going to cover a little bit of the background information that might help you to kind of read this. So when we talk about the hippocampus, there's a couple of primary areas. We're going to talk about the entorhinal cortex that's going to process sensory information both in and out. The dentate gyrus is really what's going to hold those engrams or the physical location of a memory. And then it's going to receive sensory information from the entorhinal cortex about what's going on. And those memory cells are going to activate the hippocampus, which is going to include the CA2, CA1, and CA3. And then the hippocampus is going to form this kind of loop back with the entorhinal cortex that's going to retrieve the memory from all of the like cortices, like the retrosplenial for kind of like time-based memories, the parahippocampal for kind of spatial memories, and the perirhinal for these other association areas. And so when we're targeting the dentate gyrus, that means that we're targeting the physical location of the memories because that's where it's held. So if we go back, you can remember that they are manipulating memory engram bearing cells. So they are manipulating the cells, they are changing their activity, and those cells are found in the dentate gyrus or the CA1. Okay, so what kind of we're left with is they are going to optogenetically, which we're going to discuss in just a second, changing the activity of these cells to see the effects, and those cells are found in the DG or the CA1. Okay, so on to optogenetics. Optogenetics is kind of it's a really popular and super cool technique in neuroscience where you're going to implant special ion channels, and those ion channels are going to be opened by specific wavelengths of light. There are two primary ion channels that we use. Those are channel rhodopsin, which is used in this paper, and halorhodopsin, which isn't. The channel rhodopsin is excitatory. When it opens, because it's exposed to blue light, it's going to let in sodium ions, right? Kind of the same way that a glutamate receptor would, or an NMDA channel. And then halorhodopsin is going to be inhibitory. It's going to inhibit the cell by letting in chloride ions whenever it's exposed to yellow light. And so what we get with that is they are going to optogenetically manipulate memory engram cells. And what they're doing is they're using channel rhodopsin 2, that's the excitatory channel. So they're going to manipulate the memory engram-bearing cells with excitatory channels. They're going to activate these cells in the dentate gyrus when they're exposed to a particular context. So they're going to activate these cells in the DG when they're exposed to a shock. And they're going to see if, even if the animals didn't experience the shock in the neutral context, if activating the excitatory memory is going to kind of allow them to form that fear memory with the original context. Okay, we're going to move on to reading the actual introduction. So I want you to find 3 important prior research results. You're going to highlight those in blue, and then the central question and hypothesis, and highlight that in green, and then unknown terms or jargon in red. Yeah, the channels are implanted genetically, and we're going to talk about that when we get to kind of the methods section. And then they do activate the channels with the blue light. So we'll talk about all of that, and you'll kind of see how they use it when we get to the methods section and the results. Ignore this little blurb about page 3. I'm using a different PDF from you. It's just the first 2 paragraphs of the paper.
Q&A.
Yeah, we'll talk a little bit about it. So, when we're talking about a fear memory, that's really going to be the connection between a fear stimulus and the amygdala that produces a fear response, right? So, like a shock is going to activate the amygdala and make the animal feel fear. But with classical conditioning, we can create those same kinds of memories for stimuli that shouldn't normally, like a bell or a certain context. And so when we're creating a false fear memory, we're really meaning that it's going to, like, you didn't have the connection before, but we're going to do something other than exposing the animal to a foot shock in that context that's going to make them afraid of the context, as though they were shocked. Okay, are we about ready to talk about it a little bit? Yes, no, maybe so? Oh, there's mine. This cat is saying hi because it's the introduction. Okay, so go ahead and type in chat or just talk out loud. What are some 3 things that you thought were important prior research? Damage to the hippocampus impairs episodic memory. Yeah, that's good. Yeah, the population of granule cells in the DG that are contextual memory engram cells. You're good, Wordle. Yeah, right. Humans have a rich repertoire of mental representations generated internally by processes such as conscious or unconscious recall, dreaming, and imagination. That's definitely true. That also applies to when we're looking at like Alzheimer's and that kind of thing, because those are some of the things that you actually lose. So it does have clinical relevance. Okay, yeah, those are all really great. Um, So when we're really reading through a paper, it's important to note, and this is probably the most important thing I'm going to stress today, is that not everything, and actually most of the paper is just going to be completely useless when we're doing a normal read through. So that was a long introduction, right? It was 2 paragraphs long. It'd take up like a page and a half. This is all I really thought was important for this paper. I thought it was important that they identified a small subpopulation of DG cells that were contextual memory engram cells. So they're localizing where the context memory is that allows them to find it so they can manipulate it. And that optogenetic stimulation of these cells can activate behavioral recall. So this is basically just if we activate the cells, it's basically the same as the animal remembering, right? So this is really what I thought was only important for understanding the rest of this paper. The rest of it was just, it's great for leading up. And remember, the papers are like stories. And so that's kind of how the author has to guide your attention and guide your thought process. But when we're actually reading a paper critically and reading it for information, very little of it is going to be relevant. And the stuff that's relevant is usually what's in the abstract. Remember, we're going to let that guide how we read the paper. And so if we go back to the abstract, we see those two things, right? Dentate gyrus activated by channel rhodopsin 2. So memory engram bearing cells in the hippocampus. Those are the two things that were in the abstract that we looked for in the introduction. And it turns out those are really the only two things that are important. Okay, I want you to go ahead now and put what you think their central hypothesis is in chat. Yeah, that's right. I thought so too. Of course, they just say it. The hypothesis is whether artificially activating a previously formed contextual memory engram while simultaneously delivering foot shocks can result in the creation of a false fear memory for the context in which foot shocks were never delivered, right? So this tells us what they're going to do in the paper. They're going to try to create a false fear memory for the context in which foot shocks were never delivered, okay? So they're going to put the animal in a context. It's just going to sit there for a while. It's going to form a memory. Then they're going to take it out and they're going to artificially activate, remember with optogenetics, at the same time as delivering a foot shock in a different context. And if we think about classical conditioning, where we're pairing the bell with the food, right? That's going to pair them together and create that new memory that's associated with the food. In this case, it's just a fear memory, right? But we're doing that by activating the cells themselves. Okay. And then this is the kind of also what they did. This is a second part of their hypothesis. We investigated whether a light activated contextual memory in the DG or CA1 can serve as a functional conditioned stimulus in fear conditioning, right? So that's just what we were talking about. Fear conditioning associating a neutral stimulus, in this case, the light activated contextual memory in the DG or CA1 with a foot shock, right? Okay, any questions or jargon that you wanted to cover before we move on? Why did they choose DG and CA1? That's a good question. So if you remember in the introduction, this was the prior research. They actually identified that the DG and the CA1 in the hippocampus is where you find those contextual memory engram cells, so the physical location of the contextual memory. So that prior research kind of said, hey, this thing is here. If we activate it, can that like be the same as reactivating the memory? Okay. Okay, so the methods section is kind of exactly what the scientists did in their experiments. Most people that you'll talk to that are in academia, either like as undergrads or as PhD students or people beyond it, like PIs, that kind of thing, most of them will tell you that they do not read the methods section unless they really need to. Most of the time, when you have to read the methods section, it's because you are confused about something in the results, or you are using it for replication, right? You're going to do exactly what they did for your own experiment. But for the most part, we don't actually read the methods. In some cases, the authors are really nice and they make a pretty little picture for us to follow along just to kind of get a short and sweet form of the methods. But for today, we're going to skip the methods actually and just kind of talk about it in general because this can be a little confusing if you aren't in the field. So the primary method that they used was they genetically implanted this this gene for channel rhodopsin 2. If you remember, that's the excitatory optogenetic channel, right? So that's going to be active whenever we're shining light, and it's going to let the ions inside and depolarize the cell and cause it to fire. Yeah? So we can basically, with this, control when a cell fires. So the system that they use is a tetracycline response element, which is going to respond to this tetracycline transactivator. So the mouse itself had this gene implanted at birth, okay? CFOS is going to be a gene that is going to be active and it's going to produce the proteins downstream whenever the neuron fires, okay? So whenever the neuron fires, it's going to produce this specific gene called the tetracycline transactivator. You don't need to remember that. And this tetracycline transactivator is going to activate this gene that they implanted into the hippocampus with a virus. Why would you skip the methods section when the methods could make or break a study? Just because whenever you're really like reading a paper, they're already going to include that in the results. And I don't really do a deep dive into the methods unless I'm like specifically looking at why they did something or specifically how they did it to kind of replicate it. It's just not super important to me when I'm just trying to get the results of the paper. So whenever the neuron is active, it's going to produce this protein that's going to activate this, and that's going to transcribe and produce this channel rhodopsin, which is going to let us activate the cells whenever we want, and mCherry, which is going to let us see where we like which cells we tagged with immunofluorescence, which you're going to see a little bit later. And then we also used this schedule. So when they were on docs, they didn't have any of these tagged or activated neurons. They were taken off doxycycline so that they could kind of form this contextual memory. Yeah, exactly. You just hope the methods are reasonable given that it's published. You kind of trust them to a certain extent. But you always like kind of validate it. Usually, in my experience, if a paper is really suspicious, you will be able to see it from the results, right? You don't have to go digging around in the methods to find that out. But yeah. Oh yeah, no problem. Thanks for coming. So they were on doxycycline for most of their lives. And then they were taken off doxycycline for just a little bit in this A context, right? In this one specific context. And that let them form these memory engram cells. Yeah. So we're looking here in the dentate gyrus. These cells have developed into memory cells for context A. Then we're going to put them back on doxycycline and we're going to give them a foot shock. And that's going to create these fear memory cells, right? This is going to be the memory for context B, where they were shocked. And then we're going to run them back into context A. And we're going to see activity in those memory engram cells for context A again. And then put them into a new context where they have different memory cells. Yeah? So those are this is kind of the methods that they use is they're going to use this ABA-BC or sometimes AB ABBA. It's just they're going to use this same kind of condition where they first expose them to the context and let them make those memories. And then they're going to expose them to the foot shock while they activate with light these memory engram cells. And then after that, we're going to do testing to see if they have a fear response. Was there a control group with no foot shock? I Yes, actually. Well, they didn't do a control group with no foot shock, I don't think. It was just a control group where they didn't use the optic reactivation, and then they had the sham surgery, right? Because if you do everything except for pairing the two stimuli and then you put them back in A, and you see the group that didn't have the pairing is like they're scared of context A for some reason, then that would be a confounding variable, right? You'd be able to see like, oh, it was just the fear conditioning itself that made them scared of just being anywhere or scared of seeing the experimenters or something like that, right? You don't really need to run a situation where you don't give a foot shock. Okay, so for this, because it's really important, we're going to have a discussion question. So they put them on doxycycline. Why did they put them on doxycycline? Why do you think they put them on doxycycline? If you need to look up what doxycycline does, you can do that. It's an antibiotic. Does anybody have ideas? It's okay to be wrong. Wow, you are on it today, Quantum. Yeah, so doxycycline is going to bind to this tetracycline response element and block the production of channel rhodopsin 2. So as long as they are on doxycycline, they aren't going to produce any of the ion channel. So when they're in condition B, they aren't producing these. These cells aren't going to be activated by light in the future if they choose to reactivate them. And then in C, they also aren't going to produce cells that are activated by light. And so the only cells that should have this channel rhodopsin 2 in the mCherry tag are going to be the ones that are for context A. Good job. Does anybody have any questions about the methods before we kind of move on to the results? It's okay to have questions. No? Okay. Okay, so the results of a study are going to be the results of, yeah, this doxycycline defines the timing. So the results of the results of the study are going to be really the results of their statistical analysis. It's really just going to be like what they found in the study. This is when we read a paper. This is most of the reason why we read the paper is just what did they actually find, right? For more experienced readers, you can usually get everything that you need from the figures. Um, Whenever I'm just speed reading a study, I basically just read the abstract, read a little bit of the introduction. I might look at the titles of like the subheadings in the results just to kind of see what in general they found. And then for actually getting the information out of a study, I look at the figures and I look at the figure, like the boxes, the captions for the figures. And that usually tells me everything that I need to know. I only really do a full read through of the paper, like the methods and the full results, if I'm trying to replicate it or I want to get some specific like information that I couldn't get from the figures. Okay, so I want you to read the results section and highlight the central question that they're asking in green, the methods that they use to answer that question in yellow, the results in pink, author's interpretation in purple, and any unknown words you don't know in red. Let me see where the results really start. Okay, so the results section is going to start at the 1, 2, 3, 4. The fifth paragraph is where the results section really starts. It's a weird paper. I have in my version of the paper, there are some subheadings. I don't know where this version of the paper came from. It's just on my Zotero. I probably used it a couple of years ago to read this paper in class or something. Um, it's just the fifth paragraph. This paper is just weird. Yeah, you're definitely right about that. So, the internal memory activation and the real experience, like I said, they really don't kind of test that. They're kind of just testing the pairing between them. And if they don't get paired, like that itself is a control. And then for the precise hippocampal region, that's why they have the CA1, which is what we're going to see later when we talk about the results. The CA1 is their control to make sure that they're active, like actually activating the DG, and that's what matters instead of just activating any part of the hippocampus. Okay, did everybody have enough time to read through the results, or do we need a little bit more time? Okay, we're going to go ahead and start going through. And we're going to do it, we're going to get a little bit of practice with reading figures. We're going to start with this. So you probably saw this figure. This is the very pretty picture of the brain that they did after the staining. The red is going to be this M cherry, and the green is going to be CFOS, right? So the M cherry, those were the cells that are specifically the engram cells, the physical location of the memory for context A. And then the green cells are going to be CFOS. It's going to show you which neurons were active in the second context they were exposed to, right? And we have that in both the dentate gyrus here and in the CA1 down here, right? So tell me a little bit about these results, right? Why would we have these results? Why is it important to show that there's a difference in the overlap between CFOS and M. cherry when we first expose this animal to the A context? Oh, don't see that. To the A context and then to another context. Why is it important that the animals that were first exposed to the A context and then the A afterwards is different from the animals that were exposed to the C context second? It might be showing the difference between the DG and CA1. That could be an interpretation. But then they would have the DG and CA1 on the same graph. These are two separate graphs. So why are they really comparing this paradigm, the AC and the AA? Is it showing the strength of the classical conditioning response? Not quite. We're doing a little journal club paper reading workshop, Tony. How the DG and CA1 are affected per context. What do you mean by how mCherry and CFOS are tying? Oh, tying in. That's a good idea. So this figure basically is just one of their controls. They are trying to show that the neurons that they tagged in their original context with mCherry are the same neurons that are reactivated when they put them back into the context. And so we see that the overlap between neurons that are tagged with mCherry for context A and active neurons for CFOS is much higher than when they were put into context C afterwards, right? So this is basically just showing that they tagged the right cells. And we can see this in both the dentate gyrus and the CA1. There was much more significant overlap between CFOS and mCherry when they were exposed to context A and then context A than when they were exposed to context A and then context C, which if you go back to this is what we expected to happen, right? These cells are going to be different from these cells, right? But if we put them right back in context A, this cell is going to be the exact same as this cell, right? They should be activated the same. If you know about place cells, this is basically the same concept, right? The same cells are going to fire when you're in a specific place. So when they are in this place, these specific cells will fire. And when they're in a different place, those cells shouldn't fire. Instead, different cells should fire. Okay, so this is basically just saying, yeah, we tagged the right cells. We know for a fact that these are context A cells, and when we activate them, we're going to force the recall of context A. Okay, so tell me a little bit about this figure. Let's start with F, and then, yeah, let's just start with F for now. So tell me a little bit about what these mean. Remember, we just talked about this in the methods section. What do the dotted lines mean? Oh, Lord. I think those might just be averages. Hang on, let me look. Was that figure 2? That was figure 2. So these are the dotted lines are going to be the what's it called? The error bars. So those are the chances of overlap. So when those are separate, it means that it's statistically likely that this isn't due to random chance. Or it's very statistically unlikely that it's due to random chance. Yeah, it's like a confidence interval. Yeah, so the result for the DG is more statistically significant, and that does kind of make sense because that's what you'd expect. The CA1 should, like, that's the output, right? It shouldn't be associated with the physical location of the memory. But the DG, that's what you'd expect to find the difference between context A and context C for the actual memory. Yeah, right, because it's where the memory is. Okay, so tell me a little bit about this figure, right? So we have the freezing here in the DG and the CA1. Tell me about what they did in figure F. Any ideas about what's going on? Oh, Sona's talking. Yeah, so they are going to, yes, exactly. So they're going to start with exploring the safe environment, right, in channel A, and then they're going to be exposed to the foot shock while activating the neurons with light. And then they're going to put them back in context A, and then in a new context, context C, to measure their freezing levels, right? So this A here is going to be the initial freezing. This is going to be the new context A after the pairing. And this is going to be context C also after pairing, right? And so what really do we see in this? What are the major results just for this first figure? Right, yeah. The mice with the channel rhodopsin, right? That's the ones that are optogenetically activated. That's the ones where the context A was paired with the foot shock. Freeze significantly more when they're placed back into context A, right? Specifically when it's in the dentate gyrus that those are activated. And then for the CA1, we don't actually see a difference when those cells are activated. And that's because we're activating the physical location of the memory. Yeah. And so this basically just shows that they did actually create a false memory where when they activated the DG cells, even though they'd never, like, this isn't a dangerous context, context A isn't a dangerous room in general. When they place them back into there after kind of pairing it with the foot shock, we see an increase in freezing. Yeah. Okay. Then for this one, they do the same thing, but they put context C right before the foot shock pairing, and then they move context C to before context A. Yeah? So give me kind of an idea. Why do you think they would first expose them to context C and then do the foot shock pairing and then expose them to context C again? Why would that be important? Maybe to check which one would create a false memory. Good idea. If there was equal activation for A prime and C prime, it would serve to make the explanation less likely. To potentially provide proof that it's the foot shock that triggered it. Yeah, exactly. It's to see if the freezing would transfer to context C somewhat. So what we're really looking for, right, exactly. Salience got it. We're looking to make sure that initially this change in freezing here wasn't because this is a novel context, but they're really looking here to make sure that it's not because C was the first context they were put in right after B. Because if you see here, they put them in context A right after they paired the foot shock, right? So here they add C before they put them back in context A, just to make sure that it's not just because that's the first context they were placed into after the pairing, right? But we can still see that the mice that were optogenetically reactivated had more freezing than when they were put in context C. So it can't be just because it's the first context, like relevant context they were placed into after the pairing. It's because they were actually reactivating the neurons that were coding for context A. And we can kind of see that same thing with this new control that they added, where they used the same mice that had the channel rhodopsin and mCherry. So these are the neurons that are tagged as being concept A, and they have the channel. They're just not activating them with the light. So they're showing even here that, hey, we did the entire procedure, and it's this specific part where we activate the light and activate these memories that causes the pairing of the fear memory, right? So that's just another control that they did to show that it was the specific activation of the memory at the same time as the pairing of the foot shock that triggered this change in freezing behavior, right? It's not any of the other things that they did before. Okay, any questions before we move on? The figures are a huge learning curve. It's the figure captions help a lot because they usually tell you exactly what they did. So if I'm ever having trouble understanding a figure, I usually go looking at the figure captions, but this is definitely one of the most important things to learn how to do is to interpret a paper just based on the figures. Um, that's because it's a lot more unbiased that way. If you can look at the figures instead of because when the authors are writing the results, I've written a results section before, they throw in their interpretations and it's really hard to tell them apart from their actual results. But when you're looking at the figures and you're just seeing like the graphs and statistical analyses, it's a lot easier to be unbiased when considering the results of the papers and like did they actually find what they said they found? Like, does this actually mean anything? Are the significance levels high enough? Do these graphs just look weird and untrustworthy? Um, so it's also a lot faster than just reading like the results sections are usually like pages and pages long. It's a lot faster as well to read the figures. Okay, we're going to move on to the next set. So they are doing this same thing, but they are going to uh, they're just going to add in context B and the activation with the light, right? And then they're going to add in a context D afterwards and also measure freezing in both of these. So what are these two graphs showing? Let's start with graph A. Yeah, so the no light means that they hadn't been exposed to the light beforehand. They were not exposed to the light when they were in context B, right? Right here. And then they're also not exposed to the light when they're in this new context B. Yeah, that's a really good interpretation, Sona. That's actually right on. So, oh, not that. So when we're looking at this D is a context that they've never been placed in before, right? And we should see that whenever they are placed in this context, they don't have any fear, which is what we see in the no light, um, this no light group, right? They don't have barely any fear. They might have a it's a new context, just tend to freeze sometimes. They just do it. But there's a really low amount of freezing. But the second we turn that light back on in these mice they were in that context and they had that pairing, their freezing in like skyrockets. It's really a significant increase, right? So that shows that we can activate this memory even in new contexts, right? It isn't just them being in context A specifically that causes the light. And it's also not just being in a new context that does it. This difference is really because we activated this fear memory that was created, right? And then here we can see that there's actually a crossing interaction, which is really important because it shows that both the context and the light are really important. So we can show that, or sorry, the presence of the light before and the presence of the light now are really important. So this is really showing that effect of activating the light while we were in context B is what's triggering this stronger increase in freezing, where the other one is decreasing in freezing, probably just because they're getting used to being in the context and realizing, hey, I'm not getting shocked here, right? Okay, so we're going to move on to this part of the figure. So does anybody remember what green means? What does the green tell us? Yeah, the green is CFOS. You're right. And what does the CFOS tell us? Yeah, that's a good point, Salience. They are differently scaled. And so what that would really tell is I'm not too worried about it because they have two bars of significance, and that's like p is less than 0.01. So that's pretty strong. But if it was like like a single bar of significance, that might indicate that these results aren't necessarily super statistically significant, right? Or that this graph might not be trustworthy. And I've actually seen that happen before. I've read a paper where the bars were scaled differently, and I didn't realize it at first, but their the results weren't that significant. And when I was going over it with a professor in like in after the fact, he was like, "Hey, I noticed that these were very differently scaled, and so these results are a little suspicious, right? They might not be as significant as they're trying to make it appear." So the CFOS is really going to tell us that these neurons were active, right? These are going to be the neurons that are active during, in this case, it's going to be in natural recall. In time B, this is going to be for artificial recall when they activated that memory back in context A, which should be the neutral stimulus. And then here in a completely different context, we can see there's almost no CFOS activation. Yeah. And so what are these results really telling us? Yeah, right, exactly. So when they activated this false fear memory, it's going to activate the same regions as activating the natural fear memory, right? And that's going to be significantly different from just putting them in a new context, right? Okay, I think this is our last section. So tell me a little bit about what they're doing here in in A. Any idea? Yeah, so this is going to be what we call conditioned place avoidance. So before, they were just put into this neutral context, taken out and put into the fear context, taken out and put back in here. Instead, with this, they're just allowed to pick which one they want to go into. And so we're going to create a memory for context A, this room over here. Um, and then we're going to let them kind of wander around for a little bit. They're going to get to go into this new context without the memory, where the memory isn't created. And then we're going to pair the light with the foot shock again. and put them back. And we're going to let them choose which room they want to spend their time in, right? And what we can see over here is that when it was just the cherry, right, just the label, they don't have the light activation, right? There's no pairing of context A with the foot shock. They spend their time roughly equally in both rooms, right? There's not a super big difference between them. There's not a big difference between where they choose to spend their time. But when we label the left chamber, you can see that this is where the fear memory is for, or the false fear memory is for. They spend way less time in here than they do over on the right side, right? They spend way more time on the right side, which you can kind of see with the heat map. And then when we flip them, right, they're labeled here. This is where the fear memory was created. They spend way more time on the other side, yeah? And so we can really see that in the difference score. When they have the light activation and the creation of the fear memory, there's a much stronger difference score in where they choose to spend their time than just over here with just the labeling, right? So this really shows that there's an effect of the activation of the fear memory. It's creating a strong difference in where they choose to spend their time. And if we see this over here, there's a strong increase in chamber preference for, in this case, this is for the labeled chamber, or sorry, the unlabeled chamber. This is for the unlabeled chamber. There's a strong increase in preference for the unlabeled chamber than for the labeled chamber, or sorry, than for the just the mice that didn't have the activation, right? So this is showing that the activation creates this fear memory where they want to avoid the context, the chamber where the fear memory was created. Yeah? Okay, so we're going to move on to the discussion. Does anybody have any questions about the results of this paper? I'll run through them real quickly just so that we can kind of get all of it. So there was a strong overlap between CFOS and mCherry that showed that they labeled the right cells, right? So they were working with the right cells. This wasn't just because they were just activating the hippocampus willy-nilly. It's just because specifically they activated the dentate gyrus cells for context A. For this, they showed increased freezing in context A after the pairing, and they didn't see that in context C. And so that specifically, they saw increased freezing because they were doing this pairing and then reactivating this artificial fear memory. Here, we see that there's some crossing interactions showing that it's really important that they were both conditioned with context, with the light, and that the light is what's really causing this increase in fear in both contexts. Here we're showing that the false recall is going to activate these same areas of the brain and with very similar strength as the natural recall, right? This is like your unconditioned stimulus, and that's significantly different from this neutral context where there's no fear memory. And then here we're going to show that they have preference for a chamber that doesn't have the fear memory over the chamber that does have the fear memory. And this is different from when they were unlabeled, right? There's no preference here. There's very, very little preference. Okay, so I want you to restate their research question. Yes, I can. That was a while back. So the research question is whether artificially activating a previously formed contextual memory engram, that's for context A, right? They formed that contextual memory engram while simultaneously delivering foot shocks. That's really what they're talking about in this context B. They're activating these engram cells for context A while they deliver the foot shock. And they're seeing if that can result in the creation of a false fear memory for the context in which foot shocks were never delivered. So they're seeing if that creates a fear memory so they have more freezing or avoid the context A, which was paired with the foot shock. Okay, so the discussion is primarily for providing a summary of the results. It's also going to provide the author's final interpretations, why they think these results are important, like why they're actually publishing this paper. And it's also going to provide some future directions. So we're running a little bit low on time, so I just want you to give me one future research direction that you can come up with. I'll give you all a couple minutes. Yeah, good. So, doing additional work to look at the CA1 engrams instead of the DG. They did focus a lot on the DG, um, using this model to study neural mechanisms behind memory errors in humans. Yes, that's definitely something that we could study in the future. Maybe if you could see how reward conditioning is related to this, that's a good point. This was just fear memory instead of reward memory. So if we had like the same, like we gave them a drug in this specific context, could we kind of do the same thing? That's a good point. Or like for like a light, right? We can use a light as a bell instead. So what if we use the activation of these neurons as kind of that stimulus to get them to press a lever for a drug? That's a really good one. Okay, well, that is really all I have. We're going to do another one of these in a couple more weeks. It's going to be over a paper that's a little bit harder. Um, I'm going to send it out probably 2 weeks in advance just so that you have time to actually read through. But your homework in the meantime is to try to read one paper a week to practice. I always read usually a paper a day, honestly. So I will post whichever paper I pick to read each week. We can have maybe a little mini discussion, or if you just need ideas. But your homework is to practice reading one paper a week until we do that intermediate workshop. And we're kind of just going to do the same thing again, but it's just going to be a little bit harder to understand. So you'll probably want to spend a little bit of time with that paper before we actually do that second paper discussion. Definitely don't be afraid to struggle. The more that you do this, the easier it gets. This was probably the biggest learning curve when I was first getting into neuroscience was reading papers just because it's so different from anything else that you really do in science. Um, if you need help with reading a paper, you can always DM me. Um, yeah, good job today. You guys did great. Does anybody have any extra questions before we leave off for today? I'm going to send out the PowerPoint just in case you need some help with kind of how to read each section of the paper that you pick. So just let me know if you have any questions. You can always at me in Lounge. I'm usually lurking. Alrighty, yeah, I will add the next workshop to the events planner as soon as we figure out when we're going to have that, and I will send out the paper 2 weeks before.